Good morning, everyone. Buenos días. We welcome you to our first ever Latinx BME Symposium. I am Dr. Ana Maria Porras, and this is, like I said, our first symposium ever, which we are co-organizing today in partnership with the Biomedical Engineering Department at Florida International University. And we are all very, very happy that you decided to join us today. I can see in the list of attendees a lot of familiar names, but also a lot of new names. So hola, bienvenidas y bienvenidos. And now I'm going to hand it over to my Latinx BME co-founder, Dr. Brian Aguado.
Good morning, everyone. Buenos dias. Thank you so much for joining us today. We're just so delighted to have organized this symposium with Florida International University, and we're just thrilled to see everyone joining. I will kind of kick everything off and start us off by presenting a few slides introducing our community. So if you just give me a moment, I will share my screen. So once again, welcome to the first Latinx and BME Symposium. We're just super thrilled to have collaborated with faculty at FIU and getting this symposium up and running. And like Ana was mentioning, so Ana and I co-founded Latinx and BME. Please feel free to follow us on Twitter. And Ana is currently a postdoc fellow at Cornell University, and I am currently a postdoc fellow at the University of Colorado Boulder. And Ana will be a rising assistant professor of biomedical engineering at the University of Florida. And I am a rising assistant professor of bioengineering at the University of California, San Diego. So we're super excited to start our faculty positions. We also wanted to introduce our two amazing organizing committee members from FIU, Valentina Dargam and Andres Peña. If you both would like to say just a brief word to introduce yourselves, maybe?
I'll go first. Hi, my name is Valentina Dargam, like Brian mentioned. Hola, mi nombre es Valentina Dargam. And I'm very excited to be a part of this and help put this symposium together. Dr. Jung invited me. She's the chair of the Biomedical Engineering Department at FIU, and where I'm a PhD candidate. And we've been working very hard to make today happen, and I hope that everyone gets to enjoy it.
Sure, thank you, Valentina. Buenos dias, good morning, I'm Andres. So I have been at FIU for a long time, and I'm really, really excited to see an event like this happening, and even more excited to be part of the organizing committee. Thank you, Brian. Thank you, Ana, for all the support that you've been giving us, and for this great opportunity to hold this event. And I'm very, very looking forward to all the sessions today.
Fantastic, Andres. Thank you. So just very briefly, I thought that it would be good to introduce the Latinx community and kind of give everyone a perspective about Latinx representation in STEM and how Ana and I built the Latinx and BME community. And then Ana will take over and talk about the FIU and Latinx and BME collaboration and how that got established. And Ana will also walk us through our today's agenda. So today, we thought that it would be good to kind of just suggest to everyone that Latinx are still underrepresented in engineering. So in 2018, the American Society for Engineering Education released the Engineering by the Numbers book, suggesting that approximately 11.4% of all bachelor's degrees and 6% of all PhD degrees in engineering are awarded to Latinx students. And furthermore, as of 2016, only 2.3% of tenure track faculty in biomedical engineering are Latinx. And this actually represents only 587 tenure track faculty in the engineering space out of approximately 27,000 faculty. And interestingly, only 48 of those 587 engineering Latinx faculty were born in the United States, which I think is another incredibly eye-opening number. Given that we aren't able to retain faculty from US-born Latinx folks. So really, Latinx at all levels really struggle to find senior and peer mentors that represent them and often lack access to a supportive community. So that's kind of why Ana and I wanted to build Latinx and BME. And Latinx and BME got its start at the Gordon Research Conference in 2018. And this is a picture of 4 of us. So this is a picture of myself, Ana, and Tatiana Segura and some other friends. And we were 4 Colombians at this conference, and we felt like we had some level of community. But then once we went home, we realized that that geographic distance was really hindered our ability to kind of bring together the Latin American community for ourselves. So what we really wanted was a space to make friends and to just build community, which is why we decided to co-found Latinx and BME. So really, we wanted to build a community of peers despite geographic distance to really feel connected with each other through scholarship and friendship. And we wanted to promote and support the professional and personal development of all Latinx and BME and increase the representation of Latinx and BME at all levels of our profession. We launched the Slack community in February of 2019. We have a variety of different channels to talk about specific topics associated with pursuing careers in academia, careers in industry, and alternative careers in science communication and government, and et cetera. And we really wanted to engage all biomedical engineers across different time zones and locations. So this Slack platform really allows us to do that. And then we can also initiate private or group messaging between members. So it's really a terrific platform to be able to bring together members across the world, really. We recently published a perspective piece in Nature Reviews Materials that describes our community. We have, as of February 2019, we have 245 active members on our Slack. And out of 57 surveyed members, this is kind of the breakdown. We have mostly graduate students and trainees in our organization, but we have people ranging from different careers, ranging from undergraduate all the way to assistant, all the way to full professors or even department chairs. And we have some folks representing industry as well. So we really have successfully built this community for all Latinx and BME that counters feelings of isolation and exclusion and really fosters the inclusion and success within the broader BME community. So with that, I'd like to pass it over to Anna, who will kind of detail the FIU and Latinx BME collaboration that is happening today.
Yeah, thank you, Brian, for that small introduction. I'm really here for this portion. If you can go to the next slide, please. I'm here primarily, I want Professor Ranu Jung to tell us a little bit more about why, how this collaboration came to be, what motivated her to reach out to me during last year's BMES 2020 virtual meeting, which is really when this idea got started. And so without further ado, Professor Jung, if you want to give a few words and explain.
Buenos días and muchas gracias is how I'm going to start the day. And thank you so much, Anna and Brian, for actually forming this Latinx in BME community. A community that can support peers, that can mentor others who are coming up along, and actually informing the ones who are trying to mentor you. So the professors, the industry folks, for all of us to understand what it needs to build this whole BME community. And in that BME community, the Latinx community, we need all of us together to be able to deliver on the promise of biomedical engineering. And for that, we need all of the voices and all of the training and all of the excitement. And so when I met Anna through the BMES meeting, she had, shout out to BMES for having arranged for this kind of, you could connect with people. And I saw there was this Ana Porras, who seems to be doing all this communication and reaching out to different people. And I said, I need to connect with Ana Porras. We graduate the largest number of biomedical engineering undergraduates, but what about our graduate students? What about our postdocs? Exactly what Brian just put up in the numbers score earlier. So it's like, I need to contact her. I have heard from my own students. You hear today are Valentina, a graduate student who came up from as being undergrad to grad, and I hope one day postdoc and maybe academia or industry, wherever she goes. And Andres Pena, who was also an undergrad, then was in my own lab as a doctoral student, and now a postdoc. So this, I see them. I've been fortunate personally to be part of it, our department is fortunate to have this opportunity to build this community of biomedical engineers. And so there was Ana Porras. And so I said, I'm going to reach out. They already have this fantastic platform. And this is a way for us to really move this conversation across the nation, support that group. And my hope is that this will become an annual event, and maybe another university, if not us, will support this next year. And this will continue to happen. So all the best for today. I'm looking forward to it. Our BME department is looking forward to it. We've already had a fantastic experience of working with you all, and congratulations on launching this whole community, this whole space, to give us a sense of belonging. Thank you.
Thank you, Professor Jung. And we really appreciate your support. Without the support from FIU, we could have had less organized and less professional version of this symposium. So we really appreciate all of the support that you and your department has given us. And we agree with you. We hope that this can become an annual event that multiple universities sponsor in the future. And so thanks again. And so with that, we're running a little bit late just because we started a little later to make sure all of you had time to come in. But I'm going to give you a little summary of our agenda for today, and we'll just get going with our talks. And so today our day is divided in two main parts. So first, we're going to have a series of scientific sessions, and then which are going to happen in the morning and the early afternoon for the East Coast, in the morning for the West Coast. And then later in the day, in the afternoon and evening, we're going to have a lot of what we call personal and community development sessions. And before I go into a little bit of more detail into that, I want to point out a few like tiny changes that probably most of you don't notice. And it's that in the, thank you, Brian, for moving your pointer. In the short talk section, we're going to show some talks for the first 45 minutes, and then we'll move right into the award ceremony, where we will be showing the top 4 talks that were scored out of everyone who submitted an abstract for that competition. And so, Brian, if you could go to the next slide. We're going to start this morning with 5 live platform talks, which were selected from 40-something abstracts that were submitted for consideration. I'm super excited to hear what all of them have to say, from Nancy Ruiz-Uribe, Carlos Ceballos González, Lilibeth Ortega Pineda, Dr. Santiago Correa, and Professor Carlos Aguilar, from a wide variety of institutions and scientific disciplines, subdisciplines. After that, we'll have our magnificent keynote speaker, Professor Tatiana Segura, who, like Brian mentioned, she was there at that GRC back in 2018 when we first started thinking about this idea for Latinx in BME. Then we'll take a small break for lunch or coffee or whatever you want to do in that time. And we'll move on to our short talk competition. We have 34 submitted talks. All of them are available on demand. And you should have gotten in the email that contained the agenda for the day and the links to join this session and the afternoon session. There should also be a link so that you can watch all of the submitted talks. They're all 3 minutes. So I'd recommend during one of our breaks, maybe, or tomorrow, this weekend, going to watch all of them. It's super enjoyable, and everyone put in an enormous amount of work. Unfortunately, because of time constraints, we cannot show all 34 of the talks. So what we did is we had a panel of judges to score every single talk. Every talk was scored by at least 3 people. And we are showing just the top 16 or so talks within those 34 that were submitted. We're going to show this afternoon live the honorable mentions that happen to fall in these 4 categories: undergraduate presentations, biomechanics, in vitro models, and biomaterials. And like I said, after that, we're going to announce and broadcast our 4 winners. And that will conclude our scientific session. But before that, and we'll do this again in the afternoon, we just briefly want to thank all of our short talk judges who volunteered their time graciously to make sure that we could have these short talks ready for you today. And finally, in the afternoon, we're going to have what we call our community building sessions. We encourage everyone to come, whether you are Latinx or not. I promise you will get a lot out of them. And just to warn you, it is a separate link from this link that you're using today. So make sure to check your emails or DM us on Twitter, or if you're on our Slack, we'll post it on our Slack. So first, we're going to have a 1-hour session on the science of self-care. followed by a panel that I will moderate on intersectionality that has 3 members of our Latinx in BME community, as well as Kathleen Warman, who has a more clinical perspective on this topic. And then finally, this evening at 7 p.m. Eastern, we're going to have a show and tell / social where we're going to be, and maybe if you want to participate, have something ready, something that for you represents your family and your connection to your family. That's a topic that we're going to be exploring, how our families shape our Latinx and BME identities. And so with that, I think all we want to say is we hope you enjoy the symposium. I just saw Orlando just posted the link in the chat, or where you can watch all of our short talks. And don't forget to tweet throughout the day and use the hashtag Latinx in BME, tag Latinx in BME, as well as the FIU Biomedical Engineering Department. And thank you. And with that, I'll hand it over to Valentina so that we can start our scientific presentations.
Perfect. Awesome. We'll look forward to today. So just as a reminder, we're going to have 5 platform talks, and anyone's open to asking a question through the Q&A chat box. Make sure to put some questions. We have each platform talk speaker has 3 minutes to answer questions at the end of their talk. So please feel free to post the questions while they're presenting or at the end of their talk as well, so that I'm able to tell them your question and then they're able to answer it. So without further ado, we're going to start with Nancy Ruiz-Uribe, who's a graduate student from Cornell University. Take it away.
Good morning, everyone. Hola a todos. I'm very excited to be here in this platform, sharing more about my research. So I'm going to share my screen here. Can everyone see my screen?
Yes.
Okay. I'm Nancy. I am a 4th-year PhD candidate at the Schaefer Nishimura Lab in Biomedical Engineering at Cornell University. And today I'm going to talk about my research on Alzheimer's disease. Alzheimer's disease is the most common form of dementia. It currently affects more than 50 million people worldwide. It is characterized by the accumulation of 2 main proteins, being the first one amyloid beta in the form of plaques in the brain tissue, and tau protein in its hyperphosphorylated form inside the neurons. And these 2 proteins will aggregate in the tissue and will start to slowly consume the tissue, leading to progressive and irreversible neurodegeneration. There is currently no cure or treatment for this disease. Research on Alzheimer's disease has been focused a lot on understanding these 2 proteins I mentioned, amyloid beta and tau. However, the research has shifted a lot during the past few years to understand the vascular contributions of the disease. And the reason for that is because baseline cortical flow is reduced in Alzheimer's disease patients and mouse models of the disease. This blood flow deficit is one of the earliest biomarkers, and it definitely contributes to the disease progression. This is because blood flow is critical to maintain oxygen and glucose metabolism, neuronal health, and the clearance of toxins and waste from the brain. However, the causes of these blood flow reductions in Alzheimer's disease are still unknown and highly debatable. My lab published 2 years ago a proposed mechanism called capillary stalling or neutrophil adhesion. And so what it consists is that there's this tiny fraction of capillaries in the brain that are plugged with a white blood cell. So you can see here, capillaries are very, very tiny vessels where only individual red blood cells can flow through. And in the Alzheimer's disease mouse model, there's a 4-time higher fraction of capillaries with stalled blood flow. When an antibody against neutrophil adhesion is injected into the mice, it's called alpha-Ly6G, the fraction of capillaries with the stalled blood flow is significantly reduced, and in a consequence, the penetrating arterial blood flow in young animals where the amyloid beta accumulation is very low, is actually increased significantly. And also in old animals where the phenotype has already become pretty advanced, there is also a significant increase in blood flow. And this is accompanied by an increase in cognitive function as well. But we don't really know where the stalls are actually coming from, and this is where my project goes. My hypothesis is that stalls are a combination between oxidative stress and inflammation. And I already told you that amyloid plaques will aggregate in the brain tissue, but they also bind to the blood vessels. And when amyloid beta interacts with blood vessels and binds to endothelial cells, it will activate certain receptors in endothelial cells, and then that will activate NADPH oxidase or NOX2. And this is one of the main sources of reactive oxygen species in the brain. So this increase in reactive oxygen will actually increase inflammation in vessels, increasing vascular adhesion molecules, therefore increasing the interaction between neutrophils and endothelial cells, increasing the amount of capillary salts, and therefore decreasing blood flow and cognition. And so I'm interested in understanding what happens when we inhibit NOX2 activity in the brain. So the way that I do this is that I first perform a cranial window surgery in the mice. And the reason why I do this is because in my lab, we use a technology that has become pretty standard in the field of neuroscience, and it's called two-photon imaging. The cool thing about two-photon imaging is that we can see all sorts of different structures in the brain while the mouse is actually alive. And it helps us understand blood flow, neuronal activity, and we can take a lot of physiological measurements. And so I use the APP-PS1 mouse model of Alzheimer's disease, which is a pretty standard mouse model. And then I will treat the mice for 2 weeks with a NOX2 inhibitor or a control peptide. I will image the mouse, and then I will sacrifice them to perform further analysis on the tissue. And I can also pick individual vessels in the brain, and I can measure the blood flow velocity on those vessels. Because our technique requires fluorescence, what we do is that we inject a fluorescent dye that only labels the blood plasma. So red blood cells look like black stripes. And then I can extract the blood flow velocity from these black stripes. So now I'm going to show you my results. After 1 week of NOX2 inhibition, we see a significant decrease in the amount of capillary stalls. These are vascular maps of the regions that I have imaged. And in control animals, you can see in yellow that there are actually a couple vessels that are stalled, while in animals that have been treated, there isn't any stalled vessels. So after 1 week, we see a significant decrease in the amount of capillary stalls. After 1 week, we also see a significant increase in the volumetric red blood cell flow in penetrating arterioles, but we don't see any difference in the diameter of the vessels. And this is important because sometimes when vessels vasodilate, that can actually lead to an increase in blood flow. But in this case, we don't see any vasodilation, which tells us that the blood flow is actually induced by our treatment. It's also possible for us to measure cognitive performance. And then after 2 weeks of treatment, I performed 2 analyses. The first one evaluates working memory through the Y-maze test. And you basically put a mouse in a maze that has 3 arms. And then the idea is for the mouse to perform an spontaneous alternation, meaning that it will go to each one of the arms one at a time. If it repeats the arms, then that's an incorrect alternation. And you can measure the percentage of spontaneous alternation. And in mice that have been treating with NOX2 peptide, we see a significant increase in the spontaneous alternation. The second test that I performed evaluates spatial memory. And so first we put the mouse in an arena with 2 objects, and then we replace 1 object for a novel one. And we can actually measure how much time the mouse spends with that novel object. And we see in treated animals that the object replacement preference score increases to the level of the wild type controls. So this suggests that our treatment can actually improve cognitive performance. I mentioned before that inflammation also plays a very important role. And the cool thing about our technology is that we can label vessels with whatever we want. And because capillary stalls can only be seen in vivo, we wanted to see whether capillary stalls correlate with an actual increase in adhesion molecules, such as vascular adhesion molecule, intracellular adhesion molecule, and many others. And so it is possible to actually label the vessels successfully. As you can see here, Texas red is our labeling of the vessels, and then with an antibody against BCAM, we can see that the vessel wall is actually labeled. And this is, here I'm showing you a couple of different vessels with all the different combinations of antibodies that we can see that can actually label the vessel wall. So this is something that can allows us to correlate capillary stalls with increases in adhesion molecules. So right now I'm showing you results that are currently a work in progress, but my preliminary data shows that in penetrating arterioles and in capillaries, there is an actual increase in VCAM1 in comparison to all the wild types and also Alzheimer's disease controls. And it's also possible for me to label this adhesion molecules ex vivo. And we have also seen that in treated animals, the amount of vascular adhesion molecule in the vessels is decreased, which suggests that VCAM may actually be a potential mediator between vascular inflammation and oxidative stress. And finally, I wanted to show you some cortical tissue slices from Alzheimer's disease patients, and we see a significant increase in the amount of NOX2 in human cortical tissue. So this basically is telling us that there is potential for this treatment to actually be used as a therapeutic target for Alzheimer's disease. As a summary of what I have talked about in this talk, by inhibiting NOX2, we see a significant decrease in the amount of capillary stalls, as well as an increase in blood flow and an improvement in cognition. And it's possible that all this is mediated by a decrease in vascular inflammation. And with that, I would like to finish by acknowledging my mentors and my advisors, Dr. Chris Schaefer, Dr. Nozomi Nishimura, and Dr. Teresa Sanchez, who's a collaborator at Weill Cornell Medicine, and the postdoc that I work with, Dr. Oliver Bracco, who has been the person driving this whole project and research, and all the people in my lab. Feel free to email me or contact me if you're interested in this type of research in imaging, neuroscience, or biology. That's my email down there, and I'm happy to take any questions.
Thank you, Nancy. That was great. I'm going to take a question from the audience from Joshua Hutchison, who commends you on your work. And he's asking, is there a point of no return? Once significant plaques develop, can we reverse the remodeling and restore cognitive function?
Yeah, so mostly I don't really interfere with plaques because all the therapeutic targets that have been developed for Alzheimer's disease are trying to get rid of the plaques in the brain, but this has shown to be unsuccessful. So the approach that we have decided to take into this is not to interfere with plaques because we know that it doesn't work. It's kind of a no return point, as you said. But we can actually ameliorate other things that are a consequence of plaques, like blood flow, cognition, or neuronal activity. So yeah, we don't really mess with plaques because they're really hard to get rid of.
Okay, I hope that answers this question. And then I have a question myself. Have you looked into the effect of your treatment and the progression of Alzheimer's disease? Is there a correlation between how well the treatment works versus how much the disease has progressed?
Yeah, so we do all of this analysis we have done on old mice, which already have like a very strong disease progression. So something that we can do to actually figure that out is that we can track animals from when they are very young and treat them consistently until they are old enough, and look into control animals that have not been treated, how that has helped the phenotype or the disease progression to like slow down, for example. But we haven't really looked into that specifically. We only know in old animals what is happening.
Perfect. I know Juan Pablo has had, Anna has told me that has had his hand up for a while. But I believe the only way to ask questions from attendees is to put them through the chat. So please, that's the only way to submit your questions, just for attendees. And we're gonna ask you one last question. I believe from Santiago Correa, and if you've noticed any off-target effects from NOX2 inhibition.
Not really. We have observed that it actually helps ameliorate inflammation overall in the brain. We see a decrease in astrocytes and microglia, which are like the immune cells of the brain. So we haven't really seen any off-target effects of the treatment. Yeah.
Okay, thank you so much, Nancy. Again, Anna has posted her information. You can contact her via Twitter or email, like she had mentioned, if you want to ask more questions offline. So we're going to move on to the next talk, and it's going to be Carlos Fernando Ceballos González, who's a graduate student from Tecnológico de Monterrey.
Hello, everybody. Thank you for this invitation. I'm going to share my screen. Can you see it?
Yes, we see it. Perfect.
Thank you very much. Well, thank you to everybody. Thank you for this invitation, and thank you for watching this presentation. My name is Carlos Ceballos. I'm a PhD student at the Alberto Lab at Tecnológico de Monterrey. And today I'm going to present to you this work I performed with my team during my master's studies, in which we used continuous chaotic bioprinting for creating spatially controlled bacterial microcosmos. As you may know, bacteria in nature are spatially organized. For example, if we see in our mouth, we can find this beautiful cauliflower-like structure comprising different types of bacteria. And if we look, for example, at the plant kingdom, we can also find there an association of different microorganisms forming beautiful laminar structures. And in any of these architectures, there are several factors influencing the behavior of microorganisms. For example, the presence of microscale gradients of oxygen, nutrients, metabolites, among others. And therefore, it would be important to try to recreate these architectures using any technique. In the case of conventional microbiological techniques, we have several options. For example, we can use liquid substrates, but as you may know, it might be difficult to induce spatial patterns in this kind of medium. On the other hand, you may use solid substrates, and specifically you may use, for example, your hands to induce spatial patterns on Petri dishes, but this may also be difficult at the microscale. Fortunately, nowadays, there are the so-called additive manufacturing techniques. And in today's presentation, I'm going to show you a very special one, continuous chaotic printing. This technique relies on the use of a kinesiostatic mixer. This mixer is usually used in industry for mixing applications. But in our case, we decided to put it inside a printhead to obtain the so-called kinase printhead. And then if you have two bacterial suspensions, for example, as in this case, and you extrude it through a kinase printhead, there will be an increasing number of striations as they go through each mixing element. And at the end, you will have a filament whose cross-section will have a specific number of striations that can be calculated according to this simple formula. One important feature of this technique is that the resolution doesn't depend on the diameter of the nozzle, but it depends on the number of mixing elements inside the printhead. So by increasing the number of mixing elements, let's say to 7, we can achieve a 10 micrometer bioprinting resolution, which to our knowledge cannot be achieved by neither other extrusion-based technique. And you can even push this resolution further using smaller particles, for example, nanoparticles. But in our case, we used bacteria for our study. As you will see, our bioprinting setup is very simple. You will have to culture bacteria and mix it with culture medium plus alginate. And then this mixture will give you a bioink. Then if you have two different bioinks, this means two different bacteria, and you load it in syringes, it is simplified here. With the help of a syringe pump, you can extrude it through a kinase printhead. And this printhead will be immersed in calcium chloride solution in order to obtain a solid filament, because the calcium chloride solution will help us to cross-link the alginate in the bioinks. So this solid filament at the end will have spatially organized bacteria, as in this case, this fluorescent bacteria. And then you will have to aseptically collect this filament and put it in culture medium for subsequent cultivation. In our study, we used 4 different kinase printheads, in which the number of mixing elements increased, as you can see here, and therefore the number of striations increased as well. Especially this one was considered as a well-mixed microenvironment because the lamella thickness produced by this amount of mixing elements is smaller than any bacteria, and therefore the bacteria here will be very well mixed. We firstly analyzed one proof of concept scenario in which a well-known probiotic, Lactobacillus rhamnosus GG, was co-cultured in the same filament with an E. coli engineered to produce a red fluorescent protein. And we performed this analysis during 12 hours. And at the beginning, the fluorescence intensity is very good. As you can see here, this is the red fluorescent protein produced by the red E. coli. And as the time went forward, this fluorescence intensity decreased, suggesting that probably this microorganism, E. coli, was dying. And to verify this, we used conventional microbiological techniques in order to quantify the viability using Petri dishes, as you can see here. And as you will see, indeed, E. coli was dying, no matter the construct in which it was, but the extent of inhibition in constructs printed with one mixing element was lesser compared to the other treatments. On the other hand, Lactobacillus rhamnosus co-culture in the same filaments were proliferating. And as you can see here, when Lactobacillus rhamnosus was containing constructs printed with one mixing element, it exhibited an stunted growth compared to the other treatments. We also evaluated the behavior of these microorganisms in liquid substrates. And as you can see here, we firstly performed monocultures. This means that each bacteria were cultivated in different tubes, and they were able to proliferate. but when they were co-cultured, Lactobacillus rhamnosus was growing, but E. coli was dying, as suggested by our bioprinted scenarios. So with this, we can state that depending on the spatial organization at the micro scale, the microorganisms will exhibit these different behaviors. And for example, if you want to test whether a probiotic will work better or not compared to other treatments for inhibiting pathogens, let's say, well, the microscale distribution will be a prominent factor, as this is suggesting us. Now, we also saw that after 12 hours of cultivation, at the shared interface between the red E. coli and Lactobacillus rhamnosus, there was a decreased fluorescence intensity compared to the intensity far away from there. And when we look at these beautiful micrographs, we found that Lactobacillus rhamnosus was invading the E. coli neighborhood. And we also found a similar trend in constructs printed with 3 mixing elements, as you can see here. So with this, we can say that depending on your approach, you may use a different number of mixing elements to induce spatial patterns and then to study what will happen in your specific scenarios. Then we evaluated another proof of concept scenario. In this case, we used an E. coli engineered to produce a red fluorescent protein and another one engineered to produce a green fluorescent protein. At the first sight, we may think that since they belong to the same species, they are going to behave in the same way, but the results are surprising. Here, we evaluated this co-cultivation during 48 hours. As you can see here, at the beginning, the fluorescence intensity for these fluorescent proteins is very good. And as the time went forward, it increases, and then it decreases, probably suggesting that here again, there was a starvation state, for example. And in order to assess the viability of bacteria in this construct, we again use a conventional microbiological technique using Petri dishes in order to quantify the viability. And surprisingly, we found that in the well-mixed microenvironment, this is the one printed with 10 mixing elements in the print head, there was a dominant strain, the red one over the green one. But when we induced the spatial patterns, as in this case using 6 mixing elements, there was not a dominant strain, and there was a dead phase after 12 hours of cultivation. And a similar trend was observed when we used 3 mixing elements in the print head. But surprisingly, when they were totally separated in the construct using 1 mixing element, there was not only both bacteria growing at the same rate, but there was a stationary growth phase for both of them. So we then analyzed the viability of these microorganisms in liquid media as well. And when they were in different tubes, they were able to grow at similar rates, as you can see here, achieving a peak growth at 9 hours. But when they were co-cultured in the same tubes, there was again a dominant strain, the red one over the green one. And this is pretty similar to what happened here. So with this, we can state that depending on your approach, you may use a different spatial organization for your particular purpose. For example, let's say for biomanufacturing applications, if you would like to have both strains growing at the same rate and having a stationary growth phase, or to have a dominant strain over another one. This is according to what you would like to do. Well, summarizing, we have used a novel bioprinting technique called continuous chaotic bioprinting to print spatially controlled bacterial microcosmos at a resolution that hasn't been reported before. And our proof of concept scenarios are suggesting us that the spatial distribution influences the growth behavior of these microbial populations. what are the future perspectives for this work? Well, we hope that any of you would be able to use continuous chaotic bioprinting to create polymicrobial systems. In order, for example, to recreate the beautiful cross-section of a tree bark that we were mentioning before. And why not to try to recreate tissue microbe scenarios? For example, in our gut, microorganisms are not in direct contact with our intestinal cells, but they are separated, and you can achieve it using our tool. All of this using the Kennex Springhead. I would like to acknowledge my mentors, as well as this fantastic team, Joana Bolivar, PhD student at the Alberto Trujillo Lab, and these fantastic undergrads, and to all the Alberto Trujillo Lab. We are open to take your questions, and thank you very much.
Thank you so much, Carlos, Fernando. I'm gonna open up the floor to Anna, actually, who has a question, and since she's a host, she can ask it live.
Hi, Carlos. I actually have a bazillion questions, but I think I can just set up a meeting with you later. But my most pressing one is, do you think, so like, for example, in the last thing that you mentioned, which is what I'm interested in, building tissue models, but of course, then those would have to be anaerobic. Do you think your technique could also work? You could make it work to build environments for anaerobic bacteria?
Yeah, indeed. Just by using a chamber for having an empty environment, we can place these filaments in there and having an anaerobic environment for them to grow. And this is only one of the applications we can find. There are other that are very interesting, and this question is very, very, very good.
Thank you. So we're going to take another question from the audience, from Santiago Correa. He says, great work, Carlos. Have you explored the influence of geometry/shape of the bioprinted materials, for example, the curvature of the constructions, on viability of these co-cultures?
This is another very, very exciting question. And indeed, well, when you mentioned shapes, I remember something that we are currently doing in our lab. We cannot create only laminar structures inside the filaments. We can create flowers, hepatic levels, and even Billy-like structures. And we haven't proved what will happen with bacteria in these spatial configurations yet, because this is a work that is currently doing by my team and I. But we hope that indeed the spatial organization will influence. There are several authors suggesting that in a specific, for example, if you allow me to share my screen, I have a slide to answer this question.
I think you may still have access. Yes.
There are authors suggesting that the spatial organization of microorganisms lead them to have access to different gradients of nutrients. And depending on the organization of the microorganisms in here, they are going to behave different, and perhaps they may acquire a competitive or a cooperative behavior, even if they belong to the same species. This is something that is very interesting for us. And there are other approaches, indeed, the use of different materials, not only for microbiological purposes. We can use, for example, GELMA to culture muscle cells, breast cancer cells, and among other materials to induce different responses. So this question is very pertinent, and thank you very much for asking that.
Awesome, thank you so much. We don't have any more time for questions, but I'm sure he can answer them offline. Again, you have his information now in the chat, thanks to Anna. So just as a friendly reminder, if you raise your hand, however, we cannot call on attendees, so just make sure to drop the questions in the Q&A box. So our next speaker is Lilibeth Ortega Pineda, she's a graduate student from Ohio State University, and she can take it away.
I'm going to share my screen. Can you see the presentation now?
Yes, we can.
Okay, so thank you very much for the introduction, and I'm honored by the opportunity to present my work today. My name is Lilibeth Ortega Pineda, and I am a PhD student of Biomedical Engineering at The Ohio State University from Dr. Hiwita Castro Lab. And today I'm going to talk about the reprogramming pancreatic tissue via designer extracellular vesicles. So the focus of this research is to explore a new avenue for treating diabetes. So there are several conditions that can lead to insulin deficiency and dependency. In a healthy pancreas, the beta cells from islets structures produce insulin to regulate the glucose levels. In a diseased pancreas, for example, during type 1 diabetes, there is a progressive disruption of these pancreatic cells that lead to a pancreatic islet atrophy and absolute deficiency of the insulin. So when the release of insulin is reduced, the glucose levels cannot be regulated. So there's also, after a pancreatectomy procedure, there is also insulin deficiency. And since the insulin is required for the glucose uptake in the peripheral tissues, these patients with type 1 diabetes rely on exogenous insulin for survival. So the significance of this study being that diabetes represent a significant burden to the healthcare system in the United States and also globally. So with about 1.5 million cases are diagnosed per year and represent a cost to the nation of around 300 billion annually. And there is a current study that projects that there's going to be an increase to 490 billion by the year of the 2030. So it's critical that we develop novel and effective therapies for treating diabetes. So the standard of care towards the insulin independence has been administration of insulin, and recently cell therapies such as the Edmonton protocol and differentiation of progenitor cells to beta cells. So, however, these approaches have different limitations. For example, administration of insulin has stability issues, need for daily injections, and continued glycemic regulation. The Edmonton protocol, which is an allogeneic islet transplantation, its widespread implementation has been limited by a lack of donor tissue and the need for lifelong immunosuppressants. And on the other hand, the differentiation of these progenitor cells to beta cells has impaired cell functionality and limited sources. So to overcome these limitations, our group has focused on designing extracellular vesicle therapies for cell reprogramming application. And this image showed a pancreas, and in green are pancreatic ductal cells, and the red dots are islet cells in which the beta cells are. So we aim to establish a safer and simpler approach to islet replacement therapies for diabetes, resolving the tissue scarcity issue by using this pancreatic ductal tissue, which is also discarded after islet isolations, and as can be seen in the image, exists in high quantities. So what makes our approach safe and simpler? The extracellular vesicles are biological nanoparticles that are released by any type of cells and that are involved in local and distal cell-to-cell communications. And recent studies have highlighted these particles as therapeutic nanocarriers because they represent a significant advantage over current delivery method, such as the lipoplexes or viral vectors. These particles are naturally derived, their biosynthesis is under immunosurveillance, and they have a high stability in circulation, and the size of the molecular cargo is not limited by the capsid side. So there are multiple ways to overexpress transcription factors inside inside the cells to release extracellular vesicles with this molecular cargo inside. However, most of the methods has been focused on viral vectors, and these methods has translational issues. So in our lab, we have been working in developing non-viral approaches for cell reprogramming using an electroporation-based technologies because they're easy to implement and scale up. So in this talk, I will specifically explain how we obtain in vitro derived extracellular vesicles. So to accomplish this therapy, we choose PNM, which is a cocktail of proinsulogenic transcription factors, means PDX1, NeuroG3, and MAFA that drive the beta cell reprogramming. So in order to obtain the extracellular vesicles loaded with these transcription factors, we electroporate human primary fibroblasts, and after 24 hours, we collect the extracellular vesicles and we treat pancreatic ductal cells. And then we study the dynamic of this EV production, the efficiency of the molecular cargo, packing and delivery to the pancreatic cells, and the efficacy of the reprogramming of these cells. So our results have showed that the designer extracellular vesicles were successfully derived from human skin fibroblasts in the order of 10 of billion of particles released after 24 hours of transfections. And as we expect, the average particle size was between 170 to 200 nanometers. And also, we saw that the PNM molecular cargo were encapsulated in these extracellular vesicles, and using a real-time PCR, we found that the level of PDX1, NGN3, and MAFA was by 500 to 1,000-fold over the sham control EVs that were transfected with an empty vector. So characterization of this extracellular vesicle content shows successful incorporation of these reprogramming factors inside the cells and also inside the extracellular vesicles at 6, 24, and 48 hours. And interesting, we also found that the number of gene copies was in higher number in the extracellular vesicles compared to the donor cells. So after that, we studied the uptake dynamic of these extracellular vesicles over time, and when they were exposed with PNM and sham-loaded EVs. In this micrograph, we can see pancreatic ductal cells, nuclei represented in blue, and the extracellular vesicles are represented in green. And as we can see, these cells were able to uptake the extracellular vesicles. And also, we found that these cells, the levels of the transcription factors, PDX1, NGN3, and MAFA in the cells treated with the extracellular vesicles was about 1,000 and 10,000 folds over the cells that were exposed with just the sham control vesicles. And we found also that the number of gene copies packed in the pancreatic cells after exposure with the designer extracellular vesicles was higher compared to the cells that were just exposed with the control EVs. After being studied, the dynamic production, the packing and delivery of the molecular cargo from the extracellular vesicles, we studied the efficacy of reprogramming of the pancreatic ductal cells. And in this micrograph, I'm showing the pancreatic ductal cells cultured by 3 days after being exposed with extracellular vesicles. And we found that the cells exposed with the PNM loaded EVs show expression of C-peptide and glucagon markers, which are hormone markers that are being present in pancreas cells. And also we see that these cells has a formation of an islet-like structure, which is also characteristic of this islet in the pancreas. And after 7 days in culture, the cells exposed with the PNM loaded EVs, in this case, the formation of the functional islet-like structure was more defined, and this cell clustering, as I mentioned, it's associated with the expression of pancreatic hormones. After 14 days, we still see the expression of both markers, the C-peptide and glucagon, and also the formation of the functional islet-like structure in the cells treated with the PNM-loaded EVs. In order to evaluate the efficacy of our method, what we want to do is compare the direct electroporation method and the extracellular vesicles mediated gene delivery. And we found that the viability when we treat the cells with the extracellular vesicles is 90% compared with the bulk electroporation that was just 10%. And likewise, we want to see the reprogramming of the pancreatic ductal cells using the electroporation-based method. And we were not able to see the islet-like formation cluster of the cells as we could using the extracellular vesicles. So these data together suggest that the engineered extracellular vesicles mediate the cell reprogramming. It's more effective and benign compared to the conventional electroporation method. So our results establish the feasibility of using these designer extracellular vesicles to improve the outcomes of patients with type 1 diabetes. And also these designer extracellular vesicle can be used as a gene delivery method of the PNM molecular cargo to induce an insulin producing phenotype. Also can be used to overcome the limitation of the pancreatic islet donors and to implement islet replacement therapies under the type 1 diabetes. These representative micrographs is an engineered islet showing the positive expression of both C-peptide and glucagon hormones markings. With this, I conclude my presentation, and I would like to thank my advisor, Dr. Natalia Iguita Castro, and our collaborators, Dr. Gallego Perez and Dr. Powell, the Ohio State University, and all of the members of the Precision Nanomedicine Labs. And I will invite you to follow our Twitter account to find more exciting results from the extracellular vesicles. I also want to thank the BME department of the FIU University for having me today, and I will be happy to respond to any questions.
Thank you so much, Lilibeth. That was awesome, and I totally agree with Anna. Those images are amazing. So we're going to go ahead and take a question from the audience, from Yvonne Lomeli. She's asking, how much time does the reprogramming last after having the cells exposed to extracellular vesicles? And how does this method compare to reprogramming using CRISPR?
Sure, this is a very exciting question. So we have been studying the reprogramming until 14 days in vitro, and as was seen in the presentation, we still see the reprogramming. The idea with future study, specifically with a diabetic model, will be to study a longer period of time, 5 months, but we have seen until 14 days so far. And regarding the CRISPR-Cas technology, I think, if I am not wrong, I think the CRISPR-Cas is using a reprogramming that could be integrated to the genome. And this reprogramming that we are suggesting is a transient reprogramming that cannot be integrated, the transcription factor that we are putting in the extracellular vesicles with the genome. But I think the CRISPR-Cas is a pretty well-defined technology for gene editing.
Thank you for that. And then we have another question for my PI, Dr. Joshua Hutchison, who I personally know loves EVs. He said, do you have any ideas about the mechanisms through which the cells internalize the vesicles?
I guess that's a big question still in the research of the extracellular vesicles, but we believe that depending on the size of the extracellular vesicles can be by integration of the membrane of the vesicles with the cells, specifically for exosomes that are being in the particle range that we are isolating.
Perfect. Thank you so much for that talk and for answering all the questions that the panelists had. And now we'll be moving on to Dr. Santiago Correa, who's a postdoctoral associate from Stanford University.
Thanks for that. Are you guys seeing my slides?
Yes.
Great. Awesome. Well, thanks to everyone who for coming, and thanks to the organizers for putting together this awesome event. My name is Santiago. I'm a postdoctoral fellow at the Apple Lab at Stanford. And today I'm going to talk to you a little bit about how we're using injectable hydrogels to provide safer and more effective cancer immunotherapy. So just as a quick background for those who might not be as familiar with cancer immunotherapy, I like to essentially explain this as having this cycle of events that we're trying to keep going to ultimately eradicate a tumor from a body. This process begins with immunogenic cancer cell death, where cancer cells release antigen into their environment, which can then be taken up by specialized antigen-presenting cells from the immune system. These APCs collect antigen, present it on their surface, and migrate to lymph nodes where they interact with T cells, which are the sort of one of the more important cells in the immune system for fighting cancer, because they're ultimately able to recognize cancer cells and destroy them. After T cells are activated, they exit the lymph node, enter the blood, navigate the tumor tissue, enter it, recognize cells that are expressing the antigen that they were trained to recognize, and destroy them. And that restarts this entire cycle. And so ideally, this keeps going in perpetuity until cancer is totally eliminated. However, cancer is quite adept at breaking this cycle down at different stages through a variety of immunosuppressive mechanisms. And so what we are hoping to do in the Apple group is to keep this cycle going and support this cycle through introduction of local therapy using biomaterials like injectable hydrogels. And the way that we really see this working is using hydrogels to modulate just the environment around the tumor or its microenvironment in ways where we are reversing immunosuppression of the tumor through release of immunostimulants, or modulating the efficiency of T cell priming by having drugs drain into the local lymphatics. You can also imagine hydrogels can recruit specific immune cells to its environment through the release of specific chemokines. And today what I'll be talking to you about specifically is how we're using this technology to deliver a immunostimulatory antibody known as CD40 agonists. And this acts on these antigen presentation and T cell priming steps of this cancer immune cycle. And one of the nice things about this therapeutic is that it can essentially transform an immunosuppressed environment into an immunogenic environment. And so there's a lot of interest in it from a clinical perspective to improve the types of responses that you can see in patients. Unfortunately, there are some limitations with this drug in the clinic. It might not be totally surprising if this drug is capable of reversing immunosuppression in a tumor and making it highly immunogenic, that it could also do that in other parts of the body. And in fact, that is what happens when we administer this drug the way we traditionally administer most antibodies, which is through an intravenous infusion. And as a result, CD40 antibodies suffer from a variety of dose-limiting side effects, such as cytokine release syndrome, hepatotoxicity, flu-like symptoms. On top of this, because CD40 is highly expressed in lymphatic organs like the spleen and distant lymph nodes, you also have what's known as an antigen sink effect, which depletes antibody from circulation rather quickly. So this drug has pretty poor pharmacokinetics compared to most monoclonal antibodies. As a result, from clinical studies so far, it seems like CD40 agonism is not really effective as a monotherapy. But at the same time, some recent clinical trials are showing that it's quite essential for combination immunotherapies. And so what we wanted to do is see if we could develop a biomaterial that could address these limitations for this particular drug and potentially improve the way that it's used in clinical settings and potentially offer a way to use other types of immunostimulatory drugs through a similar approach. And so what we're doing is using an injectable polymer nanoparticle hydrogel. So this is a supermolecular material where we have reversible dynamic interactions between a polymer, in this case dodecimal modified hydroxypropylmethylcellulose, or HPMC, and PEG-PLA nanoparticles. When you mix these two components together, the nanoparticle and the polymer engage in nonspecific interactions which ultimately cross-link into a network that can physically entrap therapeutic cargo, in this case, our CD40 antibody. Here's just some pictures of what it looks like to use our hydrogel and inject it through a syringe. And so under the high shear environment of a syringe, the polymer and nanoparticle interactions are broken up, allowing the entire system to behave more like a fluid-like material that can be extruded through the needle. After injection, those forces are no longer present, and so the nanoparticle and polymer are able to reestablish those interactions and restore the architecture of a hydrogel. As a result, it rapidly solidifies, and here we're just showing how it sticks to the glass slide and resists a moderate force like the force of gravity. And so our vision is to use this material for a strategy of local immunostimulation, where we administer it next to tumors to stimulate that tissue and the local lymphatics, and thereby avoid drugging off-target organs, which is pretty much unavoidable with a standard of care right now, which is intravenous infusions. So what we wanted to determine first was how our hydrogels ultimately change the pharmacokinetics of an antibody therapy. And so to look at this, we collaborated with Gambir Lab here at Stanford to do a PET imaging study. We used radio-labeled CD40 agonists and delivered that either with a hydrogel or with a local bolus, just a saline solution injected in the same place we would have injected a hydrogel. Because of a long-lived nature of a zirconium isotope, we were able to actually track the biodistribution of antibody over the course of 12 days. Here is just a example or a representative image of the PET study at 24 hours. On the left, you can see the local bolus. These warmer colors indicate higher concentrations of an antibody. So at 24 hours, we're already seeing a lot of antibody accumulating in spleen, liver, heart, and distant lymph nodes from where we injected it originally. In contrast, when we look at the hydrogel, almost all of the antibody is still retained in the injection site and in the hydrogel itself, and we're really minimizing how much antibody is getting released to these off-target tissues. Looking at this a little bit more quantitatively, we can calculate essentially the percent change in drug exposure due to delivering the antibody as like in a hydrogel formulation. And what we see is that we see an increase in exposure in two tissues, the injection site and the tumor draining lymph node, which in this case was the inguinal lymph node. So here in these two tissues, we're increasing exposure, and everywhere else we're seeing a decreased exposure, which is the change in pharmacokinetics that we were really hoping to see. Based on that, we wanted to see if we were also altering the pharmacodynamics, and we had two main questions that we wanted to answer. Is this approach modulating toxicity of CD40 therapy, and is it also having an impact on its efficacy? And so to look at that, we decided to take a look at what's going on with cytokines, which are a critical signaling protein between immune cells. This, by looking at this in the systemic circulation, we'll get a sense for how toxic our therapy is, because cytokine release syndrome is a sort of hallmark of this therapy. And by looking at it in the tumor draining lymph node a few days after treatment, we'll also be able to get a sense for how stimulated the tissue around the tumor is, which should give us insight into how efficacious this approach is. And what we saw was that hydrogel delivery is able to prevent systemic cytokine spikes. So here I'm showing you 4 cytokines that are commonly associated with cytokine storm syndrome: interferon gamma, TNF alpha, IL-6, IL-1B. And I'll just draw your attention to the blue bars here, which are the hydrogel conditions. And as we titrate down the dose from 50 micrograms to 10 micrograms, we're seeing a pretty rapid drop in the induction of cytokines across the board here. I'll also draw your attention to a black bar here, which represents sort of a clinical standard, which is a systemic dose at the maximum tolerated dose of 5 micrograms. And what we saw essentially is as we went down a dose, we're actually able to achieve lower cytokine induction than this lower dose, which is traditionally administered systemically. We're also seeing a benefit from the hydrogel compared to the red bars, which represent the local bolus administration of the same dose. And so just to compare this briefly to what we see locally, here we're looking at interferon gamma again, on the left to the systemic induction, on the right, the local induction. And what we see is that the hydrogels are able to mediate much higher levels of interferon gamma in the tumor draining lymph node compared to both the local bolus and especially to the clinical standard. We anticipated that this would probably lead to increased efficacy, and so to look at that directly, we decided to look at the low dose therapy in the B16F10 melanoma model. And so here we're just doing that 10 microgram dose, which we decided was the safest dose, and delivered it either as a local bolus or as a hydrogel. And what we saw was that a single dose of this treatment was able to slow down tumor growth in this difficult to treat model and extend survival. We are now currently exploring using this in a combination setting, and from our preliminary data so far, we've found that combining it with the checkpoint inhibitor PD-L1 is leading to a synergistic effect with local regional CD40 therapy, leading to around 40% long-term survivors with this B16 model, which is normally quite difficult to treat with immunotherapy. So just to conclude, what we've seen so far is that our PNP hydrogels are altering the pharmacokinetics of CD40 agonist antibodies. They're really helping us to focus exposure in these target tissues of a tumor microenvironment of a draining lymph node. And we're seeing that it helps to mitigate the cytokine release syndrome that is normally associated with this therapy. At the same time, the local therapies, and especially the hydrogels, are able to maintain high levels of effector cytokines in the tumor draining lymph node, even 3 days out after therapy. And we see that the hydrogels in particular can help make CD40 monotherapy more effective, and that both local bolus and hydrogels are able to synergize with checkpoint inhibition to give long-term survivors in this model of cancer. With that, I would just like to thank the Apple Lab and our collaborators at Stanford, and happy to take any questions.
Thank you so much for that. And Brian, I believe, has a question. Brian, do you want to start us off?
Sure thing. Thank you so much, Santiago, for the really fantastic talk. I was really curious about whether or not you've tried injecting your gel in a site away from the tumor. So I was wondering if an injection like this could potentially still prevent systemic cytokine spikes or reduce tumor growth, extend survival, et cetera. Just thinking about inoperable tumors, for example, where you might not be able to access and inject the gel directly next to a tumor.
Yeah, recently we've been working a bit with another tumor model, a lymphoma model, EG7, and I've been interested in looking at that. And so for that particular study, we injected the hydrogels, not necessarily right next to the tumor, but off to the side. And we still were able to see benefit to that. So I do think that probably it will depend a little bit on the mechanism of action of a drug that's encapsulated in the hydrogel, but in this particular instance with the CD40 agonist, it seems that if you can at least get things to drain to the same lymph node, which gives you a little bit more flexibility in terms of where you're able to inject a hydrogel, that you should still see a benefit.
Awesome work. Thanks, Santiago.
Awesome. So we're going to take a question from the audience, from Franklin Mejia. He said this was really interesting, and thank you for that. And he doesn't know if his question was already answered, but he's asking, how does loading of the antibody into the hydrogel affect its release interaction or interaction with target cells? Is most of the antibody diffused out of the gel, and can it perform its function from within the gel matrix?
Yeah, that's a great question, and something that we have been looking into a bit more with our vaccine work. But from what we've seen, and the PET studies kind of bear this out, is that over the course of about a week, you see the sustained release of the antibody from the hydrogel and the injection site in general. So the majority of the drug does eventually leave the hydrogel, and I think a lot of activity from a lot of the therapeutic activity is coming from drug that has left the hydrogel. At the same time, and from what we've seen in vaccine studies, we do have a good amount of cellular infiltration into the hydrogels, and that essentially the flavor of the infiltrates can change a lot depending on what you have encapsulated into the hydrogel. So we do think that immune cells that come into the gel are able to interact with whatever we've loaded into it and can become stimulated, which could play an important role in why we see the same amount of of the drug is ineffective as a local bolus, but quite has an effect as a hydrogel. That could be an important reason why we see that.
Thank you. And one last question, Alejandro Forigua is asking, have you done any in vitro model studies of the release profile and drug load of your carriers, and how does that compare to your studies in mice?
We do have in vitro models of release for these hydrogels, but it can be hard to compare that pretty controlled environment to what is happening in vivo. So we do see like much longer release in general when we look at in vitro release in our hydrogels than what we see when we're doing these PET studies.
Awesome. Thank you so much again for your wonderful talk, and you can find his contact info in the chat box. So just, I don't know if you're recently joining us or not, but we are running a little bit behind. Right now we have our last platform talk, and Dr. Carlos Andres Aguilar, who's an assistant professor at University of Michigan, will be giving this our last platform talk of the day. You can take it away.
Thank you so much, and thank you to the organizers for having me today and for putting on this wonderful event. It's really, really terrific to see this, both from being a young faculty to see this wonderful turnout, the next generation of students. So today I'm going to describe some work that has been ongoing in my lab. We are a relatively new lab. I'm an assistant professor here at the University of Michigan. And can everybody see my slides okay? If I can get a quick thumbs up or a thumbs down.
Yes, we can.
Great.
Well, actually, I see presenter view. Sorry, yeah.
How do I switch?
I see the one
that?
with your notes. I think.
Is that better?
There you go. I see your slides now.
Great. So for those of you guys who I haven't met before, I'm in the BME department here at the University of Michigan, and my lab studies muscle stem cells. And today I'm going to be talking about how to understand muscle stem cells and rewrite these programs to basically live forever. So today, I think for those of you who I haven't met, I love this tissue. It's skeletal muscle. Skeletal muscle is a wonderful tissue. It's a very broad tissue, right? It's distributed all over your body. Everything that has ever been done in history has been mediated through this wonderful tissue, whether it's terrific kind of athletic acts or wars or anything like that, to orating and expressing love with one another. It's all mediated through this wonderful tissue. It has a remarkable characteristic in that when skeletal muscle is damaged, as you can see here through these cross sections, it actually repairs itself. And today I'll talk about how these repair programs change, mainly through age, because in essence, the thing that tends to go first when we think about aging, which is something that we all do, something that is inevitable, is muscle. Skeletal muscle tends to get weaker as we get older. And as we get weaker muscle, that then leads to weaker bones. It then leads to a loss of independence, a more prevalence for things like falls. And as our society is getting older and older and older and older, there's really no therapies that we have to treat this loss of skeletal muscle or the loss of function of skeletal muscle. So right now it's a growing problem with nothing that we can do about it other than to say, sorry, we know you're going to get old. So there's two real mediators that have been profiled over the last 15, 20 years or so on what drives skeletal muscle to age. And the first of which is skeletal muscle has a lovely architecture with it, whereby motor neurons that stem down touch muscle fibers individually, and the interface between a motor neuron or an axon and what touches a muscle fiber is known as the neuromuscular junction. So this interface tends to become degraded with age, such that we can kind of fall, you can almost kind of see here, this is work by Greg Valdez, I guess 11 years ago now, that basically shows the interface here, this neuromuscular junction starts to deteriorate basically as you get old. And so you can kind of think about it as the motor neurons are giving the excitation, muscle is doing the contraction, but if motor neurons are signaling and muscle can't basically receive that signal, then they will never contract effectively. Another component that tends to get lost in age is something called a muscle stem cell. This is that little stem cell that is resident in muscle. It typically sits outside of the sarcolemma, in between the basal lamina, and you can kind of see it listed here in blue. But in age, we tend to lose these cells. We lose them both, not only in number, but also in their function. And so we wanted to ask, when I started my group just a few years ago now, was, is there a link? Is there a link between the levels of innervation and the loss of number and function of these muscle stem cells? And so we did a series of experiments that basically showed not only is there a relationship, but actually we can use that relationship to live forever. So the first thing that we did was we took a mouse that basically has red nuclei everywhere, except in that muscle stem cell compartment, which has a nuclear GFP, or green fluorescent protein. You can kind of see it here. This is a single muscle fiber where all the nuclei are red, except for this one little baby muscle stem cell, again, that has a green nucleus. Now, when you give a muscle injury to this model, you can see that there are green nuclei all along these little muscle fibers everywhere. It means the muscle is being repaired. It is being effectively regenerated by these muscle stem cells, which means they are healthy, they are doing their work. Now, conversely, when you give a neuron an injury, so when we do a sciatic nerve transection and take away a part of that motor neuron, and we basically kind of destroy the interface between the neuromuscular junction and the muscle fiber, what we see actually is a very, very different response. You see red nuclei everywhere and green nuclei just around the location where that neuromuscular junction exists. And they're labeled here in green. So this is the neuromuscular junction labeled in white. Those myonuclei that are derived from muscle stem cells are green. And when you put them together, you can see there's only really green nuclei around that neuromuscular junction. You can enumerate that and show basically that there is a very focused engraftment into that space. And this suggests basically that muscle stem cells do actually very dramatically respond to denervation, and they do so in a very localized manner. So we said, well, age, which we just talked about a second ago, loses innervation. So maybe there's a relationship that would exist between aging and innervation and a muscle stem cell response. So we take the same thing, we look at age muscle. Now we count the number of muscle stem cells that we get per fiber, and just as we expect, we have a loss in muscle stem cells. This doesn't change too much with sciatic nerve transection, just as I showed a second ago, or an injury, but an interesting thing happens that is different in age when we look at it compared to young. Namely, the number of muscle stem cells that are near the neuromuscular junction before and after an injury, it goes up a little bit for young, as we can kind of expect, because they do converge onto that site. But in age, it actually goes up tremendously. So in other words, the muscle stem cells that respond to that injury actually goes up in age, but we see that this response is fundamentally different than what we would expect in young, in part when we start to look at this junction here. So this is a good representation or a diagram of what we did. We basically cut the nerve, we tracked muscle stem cells and see how far away they are from this neuromuscular junction, and then how many of them became synaptic myonuclei. And in young, just like we showed a second ago, this is a very efficient process. They are trying to recatch that nerve. So in other words, when that motor neuron detracts and is trying to become reinnervated again, those muscle stem cells set up a very supportive environment through engraftment into myonuclei to catch that motor neuron back and to reestablish the innervation that was lost through this injury. But in aging, this doesn't happen. In other words, these muscle stem cells respond to this injury, but they don't engraft, they don't differentiate, And this promotion of reinnervation that we get when we are young is actually attenuated when we get old. Now, to really understand this, we use single cell RNA sequencing. So what we do is we take the muscle stem cells out using FACS isolation, so fluorescence associated cell sorting. We then sequence individual muscle stem cells using these FACS isolated cells, and then compare them to an online repository or something called tabula muracenis, which is basically a huge data set of aged tissues throughout the mouse life, and compared what we had to this. And we actually found something really, really interesting. And this first thing is you can kind of see there's purple and green. Purple are the young muscle stem cells, green are the aged muscle stem cells. And you can see in this little circled area here, there's a lot more green than there is purple. And that basically says that there are stem cells that we have sorted that look like they are clustering with other cells that exhibit functions with the neuromuscular junction. So in other words, these cells, which are normally all clustered very, very nicely here together, seem to have a different phenotype with them or a different gene expression program. When we take those cells out, we recluster them again, we see our muscle stem cell signatures. For those of you in the field who don't really know these signatures, it doesn't really matter, except you only see things like PAX7, MYF5, CDH15, CODL, only in muscle stem cells. And the interesting thing was they seem to share transcriptional signatures again with other cells that we normally associate with that interface, namely that neuromuscular junction. But the most interesting thing that we found from this was that they were expressing a gene called rapsin. Rapsin is basically the glue that holds that receptor on the muscle side or the postsynaptic side together. And we see in aging, those cells have a lot more rapsin than they do in young, and that's in part because the age didn't engraft. We can compare these to cells that literally decorate the top of the synapse, the interface again between motor neurons and muscle fibers, and we can see that they're a completely different differentiation, or they're a completely different transcriptional program here, where these guys are actually expressing cells or expressing factors that we normally associate with Schwann cells, so things like myelin and the like, and these guys are not. They're making and dedicated towards this muscle phenotype. We then took this one step further and said, if we give muscle now an injury, do we get that same phenotype? And then compared that data with somatic myonuclei. So these are, again, after muscle stem cells have differentiated, they engraft and become these myonuclei. So we compare the muscle stem cell response to somatic myonuclei, and we saw something very, very dramatic and actually very kind of interesting, namely that we can first track the response and the complete regenerative continuum of these muscle stem cells. So these are cells isolated right before the injury, they're in blue. 3 days after the injury, they're labeled here in green, and in between at day 7, you can kind of see that they occupy this intermediate space where a fraction of them are becoming those somatic myonuclei, and a fraction of them are actually returning back to the state where they occupied before that muscle injury. So in other words, they're replenishing that pool, that stem cell pool that existed before. And we can again track that through the expression of these different transcription factors and genes that they make. But the most interesting that came out of this was that there are myonuclei that are dedicated, just as we said before, that live right under the synapse. We call those synaptic myonuclei. And we can see that not only do these cells that we predicted didn't express or didn't engraft, they actually cluster exactly with a lot of these myonuclei, suggesting that they were fated for that synapse, but actually didn't engraft. And so they share characteristics of these genes here, but are fundamentally distinct from the canonical myonuclei that exist in muscle. So this basically let us come up with a simple model. And the model goes like this, where in youth, you have a healthy neuromuscular junction, you have some muscle stem cells that might be in proximity to that. And when we have age, basically, we are not letting these muscle stem cells engraft and keep that neuromuscular junction intact. So there is an inhibition, basically, of the differentiation of these stem cells to keep that NMJ intact. So we then just ask one simple question, and that is, if we have motor neuron degeneration that normally exists, and these muscle stem cells are trying to defend against that, if we rescue the motor neurons, does that in turn then rescue muscle stem cells? So basically the opposite question of aging. And sure enough, my collaborator and good friend here had a model to let us test just that. So for those of you who aren't familiar too much with ALS or the amyotrophic sclerosis syndrome. This was the Ice Bucket Challenge from a few years ago to try and draw attention to that disease. But in a nutshell, you basically get neurodegeneration in this disease at a very young age. And so my collaborator here had a mouse model for that. But in that model, she was actually able to overexpress synapsin and restore the neuromuscular junction and the motor neurons to prevent them from degenerating. And sure enough, actually, when we give the same injury, we can see a complete reversion of that stem cell response. We can do single cell sequencing just as before, integrate it with our data sets, and actually see a reversion towards a youthful state, indicating that there not only is synergy, they talk back and forth to one another to kind of patrol and keep this very sensitive and important relationship intact. So, what I've told you so far about today is that there is a very unique relationship, and it's actually this relationship that in essence mediates aging. And it's a fantastic new relationship because this, the models that we've used, which are lineage tracing and single cell sequencing, really let us elucidate this. But what we're doing now is taking it a step further. And that is just simply, if we can exert new functions through these muscle stem cells and deliver it to the NMJ, then that means we can basically start to think about cellular transplants to keep muscle function alive and well into RA. So what we've done is we've created a mouse to do just that, basically, where we overexpress these CRISPR transcriptional activators. And you can see this is a single muscle stem cell here. It's got, it's labeled where the nuclei are in red. PAX7, the transcription factor that is specific to that little population, is in green. The BFP reporter that tells us we have this CRISPR transcriptional activator in there. We're now using this to basically replenish my nuclei in age. So the last thing that I want to do, and this is really for the students that I think that are listening today, is how do you actually live forever, right? We're not going to live forever. We have a finite life around us, right? So one of our ancestors, right, the original, the original anti-aging guy, Ponce de Leon, he never found, the fountain of youth. But the truth is actually that you can live forever. And the way that you do that is by being legendary. And so what I mean by that is when you think about the research that you're doing and the work that you're trying to do, both as a student and as a postdoc and as a faculty, the idea that actually that you can work on important problems that will help improve people's lives means your legacy lives on for an extended period. We still talk about Jonas Salk. We still talk about the incredible advancements that they made 50, 100 years ago, in part because they worked on important problems, and that lets us keep their legacy alive. And another way to do that actually is to train good scientists with you. It's always going to be hard to do good science. If you're working on an important problem, it's meant not to be easy, but it's always going to be easier if you have good people to help you, especially good students. So that's my little ending for How to Live Forever. This is my group here at Michigan. I'm incredibly fortunate to work with wonderful students here, from graduate students and postdocs, and I'm very grateful to our funding sources. And today I talked to you about the neuromuscular junction, but if any of you are interested in some of these other programs, just please let me know, and I'd be happy to take any questions. So thank you.
Thank you so much, Dr. Carlos, for that awesome talk and advice on how to live forever. I've always wanted to, but I've never heard of it that way. So I'm definitely gonna take it into account and use it in the future. So in the interest of time, we're just gonna answer one question from the audience. It's from Ugo Giambini from UTSA, and he says, this is great work. He's asking, stem cells activate due to injury, but also stretching. I am wondering if the response of the aged stem cells to some type of passive stretching, either in animals after injury or in culture, will affect the outcomes as compared to the young cell population.
It's an interesting talk. The idea of exercise and rejuvenation, which is basically taking an aged person and making them feel young again, is a very active area of research. Now, I don't know per se if passive stretch would give you the same type of response, but it is interesting because exercise does actually promote rejuvenation of the neuromuscular junction and support these trophic factors locally that seem to help recruit other cells around that to make sure that that relationship is sustained. So it's a great question. I unfortunately don't have a short answer to it, but thank you again. So.
Thank you so much again for your wonderful talk, and thank you to all the platform speakers. Thank you for your time, and these were very awesome talks, very diverse fields. We can tell because there was a bunch of different people asking questions. So make sure to contact all of our platform speakers. You have their contact information now in the chat that Anna provided if you have any further questions or want to talk more about their research. So that concludes our platform talks, and Brian would go ahead and take us to the next session of our scientific sessions.